Okay, so when it comes to this signaling, these signaling pathways, this intracellular signaling, we're going to see that there's multiple methodologies that can be utilized. Previous lectures for this chapter, I mentioned proteins, enzymes activating each other, the whole dominoes, boom, 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 boom. Well, what it actually comes down to, instead of dominoes that physically push each other over, what we have here is a series of proteins, a series of enzymes that phosphorylate each other. This phosphorylation is literally the taking of the third phosphate off of an ATP molecule and attaching it to the target enzyme, the target protein. By doing this, by adding the phosphate group to specific amino acids, they refer to them as residues, because it's not just any amino acid. It has to be a specific one at a specific area within the tertiary quaternary structure of the protein to activate it. So we see that the residues are going to be serines, threonines, tyrosines, specific ones in a specific area, specific point within the protein. These enzymes, these proteins are referred to as kinases. Anything that's a kinase, as I said in a previous lecture, takes the third phosphate off of ATP and adds it to a substrate, adds it to something else. So you'll see phosphoserine, phosphothreonine, phosphotyrosine. The base structure, the amino group, the alpha carbon, the carboxyl are untouched. The R group that comes off of the alpha carbon, the R group is going to be the site of the phosphorylation. By adding the phosphate group to the R group of a specific amino acid residue, it's going to change the secondary structure of that protein, change the secondary, change the tertiary, change the quaternary. It goes from an inactive enzyme to an active enzyme. Here's the problem. Proteins are big and bulky. I mean, they're not as big as the cell, no, but they're big, they're bulky. They're anywhere from 100 to 600 amino acids in size. Each amino acid could be anywhere from 2 to 6 dozen atoms. This is big, this is bulky, okay? And you got to remember, everything's happening here by diffusion. So an activated ERK or MEK, we'll use those as examples. MEK activates ERK. Well, it's going to take time for ERK to diffuse through the cell, to get to the nucleus. Turns out one of these things, when it comes to diffusion, size matters. The bigger things are, the slower they are to diffuse. So if you need to get something to diffuse through the cell quickly, meaning getting that signal propagated from one side of the cell to the other, instead of a protein that's going to be going relatively slow, find something smaller. Something smaller diffuses quicker. These smaller things we refer to as second messengers. Second messengers are going to be small molecules or atoms that are small enough to diffuse quickly. Now, don't get me wrong. The proteins like ERK are going to diffuse to the cell, but it's going to take minutes to hours. These second messengers, which are much, much smaller, minutes to seconds, some of them milliseconds, to spread throughout the cytoplasm. Two great examples of this are calcium. Turns out calcium is a signaling molecule in our cells. Remember the ER, the smooth ER, one of its jobs is storage. One of the things it stores is calcium. the sarcoplasmic reticulum within your muscles, that's interacting with and in your muscle fibers and muscle cells, is where calcium is stored. Turns out we're using calcium pumps to take any calcium from the cytoplasm and pump it into the ER, into the sarcoplasmic reticulum, to store it away. Any free calcium in the cytoplasm above a certain concentration triggers responses by the cell. So pump it away, store it away, and then when it's released, it will spread, it will diffuse throughout the cell within milliseconds, seconds, very, very quickly. Another one is going to be cyclic AMP. You also see it in papers and textbook as little c amp, AMP. Cyclic AMP is when ATP, adenosine triphosphate, is acted on by adenylyl cyclase. Ends in A's, so it's an enzyme. Adenylyl cyclase takes ATP and makes a cyclic ring, as you see right here. A cyclic ring comes off of the sugar, back around through the phosphate, and the phosphate reattaches to the sugar. Cyclic AMP. Now look at this. This is what? A couple of dozen, 2 dozen, maybe 3 at most, atoms in size. This is about the size, relatively speaking, of 2 or 3 amino acids, which is, what? 1–2% the size of a protein, which means it's going to diffuse through the cell much, much quicker than a protein can. As I was just talking about calcium, calcium is the size of just one of the atoms here. So it's going to diffuse even quicker. Another one we're going to see in use is Ip3 and Dag. What happens is, you see here, 2 fatty acid chains, 3 carbon. 3 carbon means glycerol. Glycerol, 2 fatty acid chains, so that means it's a phospholipid. Here's the phosphate. Attached to the phosphate is some other group. Remember, I told you in the earlier chapter, you take a phospholipid, you add stuff on it, you get different flavors of phospholipids. This one's referred to as Ip2. What's the Ip2 stand for? Don't worry. Ip2. Phospholipase C, an enzyme that likes to cut. It cuts right there. Boom. Cuts, releases Ip2 and maintaining diacylglycerol in the cytoplasmic membrane. This right here, still much, much smaller than a protein. Phospholipase C is going to go running along the interior surface, the cytoplasmic surface of the plasma membrane, cutting these off, kind of like a lawnmower. Cut, 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 cut. And all of these Ip3s are going to diffuse, flood through the cell, interacting with other things, and beginning the cellular response.